right, so our next sense that we're going to be working on will be our sense of sight. So we're going to be looking at the human eye. So here's just kind of an overview of all the different stimuli and receptors and pathways, as we had mentioned previously, and when we had that first bit of notes where we talked about that, and kind of what those are. Okay, so we've got our vision, hearing, touch, smell, and taste there, and you kind of go through, and we'll talk about, we have already talked about the ears, and we will talk about the eye. So here is a diagram of the eye. You'll be doing something similar on your packet, but let's talk about a couple areas that we can really hone in on, okay? One of those is the lens of the eye. With the lens of the eye is going to take on light that is refracted off the cornea, and it's going to flip the image that you see. Okay, so everything that you see is transverse to the back of your eye or flipped upside down. So right now, if I'm looking at my water bottle in the back of my eye, that water bottle is actually upside down, and that picture gets turned around in your brain. And we'll get to more of that here later when we're talking about the eye. And then let's go back to the back of the eye, okay, when we get to the retina, the fovea, and the optic nerve. So here's where that image is going to be displayed into the back of the eye, which is filled with rods and cones to be able to make out movement and color and depth and grayscale and night vision and peripheral vision and all that stuff. And the fovea is the one central area where all that information is being sent back into the retina, the area that really is the center of your focal point. All of those rods and cones will send messages through ganglion cells that weave together to make your optic nerve. Your optic nerve then sends back a message to your brain. Okay, and as we remember from last unit, the right eye, crosses over the hemispheres and connects with the left eye connects with the right side. So here we are again with a more detailed look at the retina, taking a look at rods and cones and all of the other ganglion cells there that work together to pull that information out, which will eventually become our optic nerve. Okay. So one of the things that we're able to do is take a look at different wavelengths of light to get different colors. Okay. You can see the whole spectrum of wavelengths out there, many of them that we cannot see, right? Like broadcast bands, x-rays, we can't see those. So this is just the limited area that we're able to see that make up all these different wavelengths. And you can see the wavelength in nanometers that correspond with all the different colors. So when you're seeing something that is a dark violet or purple, something around the 380 to 400 range versus something that is red is going to be up around the 700 range. Okay. So we have three dimensions when we're looking at color. One is hue, the other is brightness and saturation. Okay, so hue is whether things are lights being reflected off and things are looking, red, blue, green, whatever other color. Okay, so hue is a psychological term because objects themselves do not possess color. It is just the wavelengths that are bouncing off of things that allow us to perceive color. Okay. So, for instance, if you're looking at the orange chairs that I have in my classroom, those chairs are actually devoid of color. It is the light that is bouncing off of those chairs into our eyes, and we are perceiving that as orange because that is the area in the nanometer spectrum that we see. So brightness is how light or dark something appears. Okay, the more intense it reflects the light, the brighter it is. The less intense, the darker or more dull it is. And saturation is the purity of that color. So how pure is that red versus, the actual color of it. So determined by the homogeneity of the wavelengths. Pure saturated light has narrow bands, whereas less saturated light will have a little bit wider bands. Okay? A saturated red light with no blue, yellow, or white in it, for example, appears very intense. That would be true of a yellow or a blue or a green. Okay. So here we're taking a look at some examples. This might look like an Instagram filter, but this is kind of what we're talking about. So when we're looking at saturation, or sorry, excuse me, hue, up towards the top, you can kind of see how this plays out, right? Where you've got the different types of colors, faded, not faded. You can see the brightness here in the pictures, what colors are popping out, what colors are more faded, and you can see the saturation in them as well, how purely red, blue, yellow versus a change in those colors. Here's an idea. You take the whole spectrum, we drop down that blue, that center blue, and then you see the less saturated. You can go from more saturated to the blue, less saturated goes down to the gray, okay, and you can even drop down even further with that middle saturation level and talk about brightness. These are fun, right? When you start to put the colors into these cone-shaped wheels that allow us to identify all the different aspects of it, okay? So the hue goes around this cone, identifying all the different color possibilities. The saturation is going to be from middle out. Fully saturated white light in the middle gets darker or more saturated and more intense towards the outside. And then brightness, you know, you've got light to dark there in the top. So here we are again, kind of the same idea, and you can see the different cross sections of this, okay? How they represent the different colors and how you see them change and adjust. And this is really the perception of your eye being able to understand it. And I like the diamond down here in the bottom left that helps explain it with the black to white and the brightness at the top. The hue, again, circling around the outside to include all colors. Saturation, pure, devoid of color to, you know, pure saturated color on the outside. So here's what we were talking about before with the inverted vision, right? So many of you may have glasses, right, or contacts or something else that assists in your vision. I used to, before having laser eye surgery, have very poor vision. Right? I was very nearsighted. I was, you know, about 2400. So when you're looking at what your eye does, it's going to look at an object, and then it's going to center that object on the lens to be inverted to the back of your eye. And with 20/20 vision or good vision, that picture is going to land right perfectly on the fovea for you to be able to see it clearly and make sense of it. If you've got vision issues, nearsighted, farsighted, astigmatism, etc., that will change that, and we'll get there. This is also coincidentally the same effect that we use for cameras. We take camera pictures upside down as the lenses transfer the light to the back of your eye. Okay. So you can see the picture of that leaf there going through the cornea, the lens getting transferred to the back, inverted to the fovea and surrounding areas. So the closer you get to the fovea, the more focused and detailed your sight is, and it gets a little bit further. As it gets a little bit further away, it gets a little less detailed. Okay. So here are the problems, right? Nearsightedness, farsightedness, myopia, hyperopia. So if you are normal vision, as we mentioned, you look at the top left and you see the flower being transferred directly onto the fovea to be able to make sense of it, and the retina. Then we've got myopia, nearsightedness, where you got the same picture, but it is being shot too narrowly inside of your eye. So the image is blurred and hard to make out in your eye. And then farsightedness, same thing, shot too far in the eye. So again, it's very blurry as you're trying to make sense of it. So we use corrective lenses, and you can see down the bottom, to be able to reshape the way that the lens is transferring the image to the back of your eye, okay, with an extra lens. You can see the myopia, you got a little bit flatter lens. You got the hyperopia, you got a little bit more of a curved concave lens. And this is some of the commonalities in sight as you get older. You can see much better sight on average early on. Then you get like the comfortable distance for around age 50, and then you start to see the desire to pull away to be able to see more comfortably because you're getting much further away. So many of you may have experienced this with your parents. If you ever go out to eat, and maybe your parents wear readers or something like that, and they hold their menu kind of out and like lean their head back. Well, that's what's going on, because they're getting to a point where that comfortable reading distance is starting to get further away. So they're adjusting what they're reading to meet that location. Okay. So when we're talking about the path of light that's going through, okay, distant objects, you have less bending or less curved because of the distance away from you. The nearer the object, the more curved it's got to be able to get the image to the back of your eye so you can see it and make sense of it. So now we can talk about rods and cones, right? Distribution of cones in human eyes. So cones are going to be able to allow you to kind of see the colors, right? So you've got artificially colored dots here. Okay, for two people is written as indicating the short wavelength cones that are blue, the medium that are green, and the long that are red. And the cones are going to help you be able to pick out colors and make sense of them. Okay. So we've got our rods and our cones. Our rods are going to be more focused around the fovea, okay, and cones will be more focused on the outer part of our eye, being able to help us see grayscale, movement, peripheral vision, where rods are going to help us pick up color vision. When we get you into class at some point, whether it's next week or even this week, we're going to do a little activity where we're going to identify our blind spot. Blind spot is nothing more than the area in the retina where the optic nerve leaves the brain. Okay. Because of that, there are no visual receptors there, so hence we're not able to see anything. We cover this up by having binocular vision, meaning the vision in your right eye is able to display pictures to your brain from the blind spot of the left eye, and vice versa. Okay. So as you can tell, that blind spot is roughly about 20 degrees away from the fovea, right? So when looking at this, you can kind of see the rods and the cones that are in those designated areas. Okay, so you see the large amount of cones that are at the fovea, and then the distance from the fovea over here are the rods. You see a lot further away. Some interesting things about this, behavioral consequences. Eyes work a little bit differently. So a bird of prey's eyes, hawks, owls, eagles, their fovea is actually at a different part in their eye, okay, allowing them to see down. So their fovea is at the top of their eye. So that's why they have to turn their eyes or their heads to be able to see, because they're used to flying above and having their center fovea in their eye staring down to be able to find the food that it is that they want to eat. Okay. So if we take a look at sensation and perception with this, so sensation, simple stimulation of an organ, basic principles. The eyes are taking in the information, sending it back to the brain. Sense organ, detection of external stimuli, okay, and then the process of receiving the stimuli becomes perception. Kind of that same chain that we set up last week of stimuli, receptor pathway, and then to your brain to interpret it.